Hello everyone, welcome back to my channel, Bio with Shansi. In today's lesson, I want us to start covering cell membrane and transport, which is chapter 4 of the Cambridge AS syllabus. If you're interested in learning about the structure of the cell surface membrane, the fluid mosaic model of the membrane, the different molecules that are found within the cell surface membrane, and the roles of each one of those molecules in the membrane, you are in the right place. Let's dive right into the content. Now, when you look at cells, we looked at the structure of a cell and we saw that every cell has a membrane surrounding it. Every cell has a cell surface membrane, which is partially permeable. So today we are going to learn why is the cell surface membrane partially permeable. We were also able to learn that the cell surface membrane controls movement in and out of a cell. It determines what enters and what leaves the cell. We were also able to see that these different organelles within a cell also have membranes surrounding them. Like an example, the nucleus has a membrane surrounding it. So that means that that membrane is able to control movement in and out of the nucleus. Same thing with the mitochondrion. We shall have the membrane controlling movement in and out of a mitochondrion. So one of the major uses of the membrane is to basically control exchange between the cell and its environment. But it has other roles. Apart from controlling transport between a cell and its environment, it has other roles. Some membranes receive messages in form of hormones, although we are not going to so much concentrate on that. Some membranes, like in the chloroplast, contain pigments that are used in absorbing light. So still this will be covered in photosynthesis if you learn about it to work. But in this lesson, we are mainly going to concentrate on how the membrane controls exchange between the cell and its environment. Now, before we go into the structure of the cell surface membrane, I'm wondering if you're remembering the structure of a phospholipid. Why do we need to learn about phospholipids? What remember phospholipids? Remember that phospholipids make part of the cell surface membrane. So we need to understand the features of a phospholipid. If you remember very well, we were able to learn that phospholipids are made up of what we call a glycerol. So they have a glycerol and they also have fatty acids that are attached to the glycerol, two fatty acids. In addition to the two fatty acids, they also have a phosphate group attached to the glycerol. So when you look at a phospholipid, it's made up of a glycerol with two fatty acid tails and one phosphate group attached to it. Now we need to remember the nature of the fatty acid tails. The fatty acid tails usually are hydrophobic. They are water hating. That means they cannot interact with water molecules. So it means that this part that has the fatty acid tails won't interact with the water molecules. It is always going to be repelled by water. Now we also have this side that has the phosphate. Now the phosphate is water loving. The phosphate is polar. The phosphate is hydrophilic. So the phosphate is able to interact with water. And as a result, the phospholipid is going to have two characteristics. It's going to have a region that is hydrophilic, water loving, and at the same time, it's going to have a region that is hydrophobic. Now, sometimes the phospholipid is represented using a circle with the tails attached to it. So the circle will be representing the phosphate, and these two tails will be representing the fatty acids, the two fatty acids. So this can also be used to represent a phospholipid. So you might find a structure like that that represents a phospholipid. So that is the structure of a phospholipid. Remember, a phospholipid has the phosphate, which is water soluble, and it also has the fatty acids, which are not soluble in water. They hate water. Now, the question is, when we get this phospholipid and we place it in water, how is it going to behave? We also need to understand that. If you get a phospholipid, these are the different phospholipids, they have been placed in water. When you place them in water, remember that the head can interact with water because it's water-loving. So all the phosphate heads are going to spread and interact with water. But now the tails are going to face away from the watery environment. Why? Because they are hydrophobic, so they're going to be repelled by the water molecules. So in this case, we are going to have a single layer of phospholipids made in a way that the heads, which contain the phosphate, are interacting with the water molecules, but the tails, which are hydrophobic or nonpolar, are facing away from the watery environment. So that is when you have just placed the phospholipids on the surface of water. Now, what if you dip them into the water? If you dip them into the water, it's going to be kind of different. How is it going to be different? When you dip them in water, they are going to form a micelle. A micelle is like a ball. So here they are showing us the cross-section of that micelle. And in that case, you can see that still they are behaving in the same way. The heads are going to interact with the water, but then the tails are going to get into the center of the micelle, and no water molecule will be getting to the center of this micelle. Still, when you place them in water, they can form what we call bilayers. Bi means two, layer, like in this case, you can see we have two layers. But the two layers of the phospholipids are still made in a way that we are having the phosphate facing the watery environment, but the tails facing away from the watery environment. So it is important that you understand how the phospholipids behave once in water. Always the tails will point away from the watery environment, and this whole region where the tails are facing is going to be hydrophobic. So it will also repel the water molecules, meaning water molecules won't be found in this region. But the heads, since they are hydrophilic, then they are going to be interacting with the water molecules. So because of that, the way the phospholipids interact with water, that is why we usually have a membrane made. So the cell surface membrane is mainly made up of phospholipids. Yes, it's made up of phospholipids, and it's made up of two layers, a bilayer. Like in this case, they're showing you an organelle within a cell and the membrane surrounding it. So this is the phospholipid bilayer because we have this outer layer here and this other layer. But you can see that the bilayer is arranged in a way that the phosphate heads are facing the watery environment outside, and the tails are facing away from that watery environment. And then still, when you look at the inside of this organelle, we have there the cytoplasm. Remember, cytoplasm contains water. So these phosphate heads on the inside are facing the cytoplasm, but the tails are facing away from that watery environment. So the cell surface membrane is really thin, usually about 7 nanometers wide. So this shows you the bilayer, like you can see here, how they are showing us the bilayer. We have this first layer and then this other layer. If you're liking the video so far, give it a thumbs up, and please feel free to share the video with your other friends so that they are also able to get the content. All right, so let's look at the structure of the cell surface membrane. Now, two scientists came up with a model, and they were able to describe the structure of the cell surface membrane as being fluid and mosaic. So that takes us to the fluid mosaic model of the cell surface membrane. What do they mean by fluid, and what do they mean by mosaic? Let's look at this structure of the cell surface membrane. Now, the cell surface membrane is made up of phospholipids. You can see phospholipids make up the greatest part. So the cell surface membrane has a bilayer of phospholipids. Remember, this is the first layer, and then we have the other layer. So both of them are going to form the bilayer. Now, within the bilayer, we have their different molecules. So that means that the cell surface membrane, yes, is going to be made up of phospholipids in a bilayer, but we have other different molecules that are embedded within those phospholipids. Like an example, we have proteins there, there is cholesterol, the proteins are of different types. Now, when you look at the arrangement of these molecules, the way they are arranged, they are able to move about sideways. Like an example, when you look at the phospholipids, they can move sideways. And that takes us to the reason as to why the membrane is described as fluid. Just like the way we hear the word fluid, it means that the membrane kind of flows. Why? Because the phospholipids are able to move about sideways within the cell surface membrane. It doesn't mean that they are going to leave, but they would just be moving within. So they are the consistency of a cell membrane is linked, or we could say is said to be similar to excuse me, the consistency of olive oil. Yes, the consistency of olive oil. So the reason as to why it is described as fluid is because the molecules, the phospholipids, are able to move about. So they can diffuse, but within the layers, yes, they can be able to diffuse or move about laterally within the membrane. That is why it is described as fluid. Now, we also have this other term, mosaic. So for mosaic, when you look at it from above, an example, if you look at this from above, you're going to see that it has these proteins that are appearing like a mosaic if you see it from above. So they are scattered all over the top of the membrane. If we have a protein here, another protein here, I'm just adding more proteins, another protein, another protein. So the fact that they are scattered all over the cell surface membrane as seen from above, that is how that other term mosaic comes in. So the cell surface membrane is said to be fluid and mosaic. Why fluid? Remember that fluid, we have seen that the phospholipids and the proteins can move within that bilayer. They can move in their layers sideways. And one other feature as to why they are described as fluid, the membranes, is because these proteins that are embedded within the membrane can also float about within the layer. They will not be leaving the bilayer, but they can float within, the way you see icebergs in a sea. Yes, that is how these proteins also are, because they can float about within the bilayer. That is still one of the reasons the membrane is described as fluid. For mosaic, we have seen that when you look at the proteins, they are scattered all over the cell surface membrane. So if you view the membrane from above, you will see that scattered nature of the proteins. So basically, that is the fluid mosaic model of the cell surface membrane. Now, the cell surface membrane has different molecules making it up. We have seen it has a bilayer of phospholipids. Now, within that bilayer, we have their cholesterol. We have their proteins. So some proteins can be just found in one layer. Like you can see, this protein is only in the lower layer. Others can span the whole layer. So those are called transmembrane proteins. Like you can see, it's moving from up here in the upper layer, to the lower one. So this can be called a transmembrane protein. Now, some proteins are just attached on the surface of the bilayer. Like this one is just attached on the surface. Then this one is just attached on the lower lower layer. Now, some proteins can have some carbohydrate chains attached to them, and that is going to form what we call a glycoprotein. Glyco for the carbohydrate chain, protein because it's protein in nature. So some proteins have carbohydrate chains. So this is a glycoprotein. Another glycoprotein, such a protein is going to be called a glycoprotein. Another glycoprotein, another glycoprotein. Please note that these glycoproteins are on the outside. They will not be found inside. So if you look at this membrane, you can see the structure has been labeled. This side we have tissue fluid, meaning we are looking at the outside of a cell. This side we have cytoplasm. So you never find glycoproteins or glycolipids on the inside. They're always on the outside. So a glycolipid is a lipid or a phospholipid, but for it has now a carbohydrate chain attached to it. That is why it is going to be called a glycolipid. So all these molecules are found in a cell surface membrane and they play different roles within the cell surface membrane. So we are going to be able to look at the different roles that each protein or each part of the cell surface membrane plays. So we have already seen the cell surface membrane is made up of a bilayer. So if you're told to describe it, it's made up of a bilayer of phospholipids. We have seen the phospholipid molecules can move about by diffusion within the layers. Now you could be asked to describe the arrangement of the phospholipids. How are they arranged within this membrane? Remember, we have seen that the phosphate heads will always be facing the watery environment. Like here, this is our tissue fluid with water. You can see the heads are facing that region where there is tissue fluid. This side, the heads are facing where there is cytoplasm. So the phosphate heads face towards the water environment or the watery environment because they are hydrophilic. However, the tails point towards each other. Like you can see them, they're pointing towards each other, away from the watery environment. So that creates a region of the cell surface membrane that is totally hydrophobic. So this region is hydrophobic. It's water hating. Why is it hydrophobic? Because it is mainly made up of hydrophobic tails. Mainly made up of hydrophobic tails. So this region is hydrophobic, but the surface is hydrophilic because it's made up of phosphate heads, and even this side, the surface is hydrophilic because it is made up of phosphate heads. Okay, so the phosphate heads are always going to be facing the watery environment, and the tails will always be facing away from the watery environment. We have seen the cell surface membrane is fluid. Now, fluidity of the membrane can be affected by different factors. So we are going to look at three factors. The first factor that affects the fluidity, remember, the membrane can either be much more fluid. If it's much more fluid, it means that the phospholipids are going to have more room to move about. So they're going to be moving about more, or they're going to be, moving about more basically. And if it's less fluid, it means they are not going to be moving about that much. So the fluidity can be affected by different factors. The first one is the type of phospholipid. Remember, we were able to look at the fatty acids. We have two types of fatty acids. We were able to see that we have the saturated ones, which mainly have single carbon-carbon bonds. But we also have the unsaturated, which are made up of double carbon-carbon bonds. We were able to see that when you look at the fatty acids that are saturated, for them, they are able to pack closely together. If these are our saturated fatty acids, they are able to pack closely together. If you do not remember saturated and unsaturated fatty acids, feel free to go under the section of biological molecules. You'll find the type of fatty acids there. And I uploaded a video about that on the channel, so you can look for it. You'll be able to study about the saturated and the unsaturated fatty acids. So the saturated fatty acids are able to fit so much closely together. That means that if the phospholipids making up a membrane are more saturated, if their fatty acid tails are saturated, then it means those phospholipids will not have a chance to move about so much because they are so much closely packed. There is no room for them to move. So the membrane will be much less fluid. So this is less fluid because the saturated fatty acids are so much closely packed together. However, if the membrane has phospholipids that are unsaturated, we saw that unsaturated fatty acids have a kink, or they have a bend on them. So this kink is usually going to prevent close packing, because even if I have another fatty acid tail that has come close, the kink is going to push that fatty acid tail so that I have more space. So whenever the cell surface membrane has phospholipids that are unsaturated with those double bonds, usually the fatty acid tails are not going to be closely packed. So as a result, the cell surface membrane is going to be much more fluid. Then we have the tail length. The tail length also affects fluidity. How? Assuming these are my fatty acids. This is our phospholipid with a fatty acid tails. So if the phospholipids have longer tails, it means that those tails can come in contact with the tails on the other side. And because we have that interaction, it means that they are going to be less likely to move about. So long tails bring about much more fluidity. But if the tails are short, if the tails are short, we are going to have the membrane being less fluid. The membrane is going to be less fluid because the tails are not that much interacting. So that means that the phospholipids are going to be easily able to move about much more than in this situation. So long tails means less fluid. Short tails mean more fluid. We also have temperature. Temperature also affects the fluidity. Remember, when temperature increases, kinetic energy of the molecules also increases. So usually when temperatures are high, the membranes are more fluid. But when the temperatures are low, it means the molecules or the phospholipids are going to have less kinetic energy. And then eventually the phospholipids are going to be, or the cell surface membrane is going to be less fluid. And usually cells have a way of controlling fluidity in the cell surface membrane. An example, if it's a cold environment, we know that when the temperature is low, we are going to have the membrane becoming much more less fluid. So what cells do if the membrane is becoming less fluid? Remember, the membrane stability has to be maintained. If it is becoming less fluid, something has to be done so that the fluidity is maintained. If it's becoming more fluid, still something has to be done for the fluid to just be maintained at normal. So if the membrane is becoming much more fluid, the fatty acids that are making it up can be changed. An example, if they are saturated, saturated means the membrane is going to be less fluid. So more unsaturated fatty acids can be added to that membrane if it's becoming less fluid. Because when you add there more unsaturated fatty acids, then it means that the fatty acid tails are not going to be so much packed together, and eventually the membrane will not be so less fluid. So the fluidity is maintained usually using that mechanism. So we have seen that the cell surface membrane is made up of proteins. We have seen it's made up of phospholipids. We have seen it's made up of cholesterol. We have seen it's made up of some proteins are called glycoproteins because they have a carbohydrate chain. We have seen some phospholipids, have a carbohydrate chain, so they form glycolipids. Now let's look at the nature of these molecules. When you look at cholesterol, first of all, cholesterol is a small molecule. Now, cholesterol is in a way that it also has a polar region and a non-polar region. So the head of the cholesterol molecule is polar, meaning it can dissolve in water. It is hydrophilic. That is why you see it facing the watery environment. But then the tail is hydrophobic. That is how it is able to interact with these hydrophobic tails. When it comes to the proteins, remember proteins are made up of amino acids. Some amino acids are polar, other amino acids are non-polar. So it means that when you look at such a protein that is spanning through the whole surface membrane or the bilayer, you're going to find that this region that is in contact with the hydrophobic tails is going to also be hydrophobic, meaning it is made up of hydrophobic amino acids. But this region that is interacting with water is going to be made up of hydrophilic amino acids. So same case here, we have this region outside, it will be made up of hydrophilic amino acids, even this region, hydrophilic amino acids, but this region that is interacting with the tails is going to be hydrophobic. So eventually, these proteins will never leave the cell surface membrane. Why? They're just going to be floating within that membrane. Because if this protein tries to leave and maybe move outwards, away from the bilayer, these regions that are hydrophobic will be repelled by the watery environment. So eventually that keeps the proteins within the cell surface membrane. Now, some proteins have channels where molecules can pass. We shall later look at channel proteins. But still in this case, you can see this region that is of the protein that is interacting with the fatty acid tails is hydrophobic or non-polar. But now this region that is interacting with the phosphate group and the watery environment is going to be polar or water-loving. So that is the description of the cell surface membrane and the features that it has. So we are going to look at the role of the different parts of the cell surface membrane, the different molecules. And we're going to look at 3 types of molecules. We have the phospholipids. We've seen there is cholesterol. Some phospholipids are glycolipids because they have a carbohydrate chain attached to them. And we also have their proteins. Some proteins are glycoproteins because they also have a carbohydrate chain attached to them. So let's look at the phospholipids. What are the uses of the phospholipids in the membrane? Number 1, these phospholipids, first of all, form the basic structure of the membrane. They form the bilayer. As simple as that. That is a very important role. They form the bilayer. They are going to determine the fluidity of the membrane. We saw that if the membrane has more saturated fatty acids or phospholipids, then it means that that membrane is going to be much less fluid. And if the membrane is made up of more unsaturated, then it's going to be more fluid. Yes. Now we have seen that the phospholipid has two regions. One region is polar, the other region is non-polar. Let's go back to the diagram that shows us the membrane. So these phospholipids are going to act as a barrier against polar molecules. What does that mean? If a molecule is polar, assuming I'm having an ion here, which is polar, ions dissolve in water or they have a charge, so they are polar. So assuming I have that ion cannot come and cross the cell surface membrane directly. Why? Because it is polar. The moment it tries to get through, it will find this hydrophobic core, which is going to repel it. Or if I have other molecules that are soluble in water, such as glucose, glucose can also not cross the cell surface membrane. Why? Because it is also polar. So one of the uses of the phospholipids is that the phospholipids, because of the tails that they are having, they act as a barrier against entry or exit of polar molecules. So polar molecules will not enter and still they will not leave the cell. Why? Because the phospholipids act as a barrier against their movements. So that is the other use of a phospholipid in the cell surface membrane. What else? We have also seen that these phospholipids can also be used as signaling molecules. We shall look at that later as we learn about cell signaling. But for now, just know phospholipids form the bilayer, and then they are going to act as a barrier against water-soluble substances. So that is going to prevent, that is how the cell is able to control what enters or leaves. Sorry, that is how the cell surface membrane is able to control what enters or leaves the cell. So the other molecule is cholesterol. We have seen cholesterol. Cholesterol is having almost the same kind of characteristics as the phospholipid. How? It has a head, but the head is polar. And then the tail is non-polar. So what is the use of cholesterol? Now, because cholesterol has a polar region and a non-polar region, we can still also say that it is going to act as a barrier against entry of polar molecules or exit of polar molecules in and out of a cell. That is one other function of cholesterol. So it is similar to that of a phospholipid. It acts as a barrier against entry or exit of polar molecules. One other role that is very important about cholesterol is that it maintains the mechanical stability of the cell surface membrane. What does that mean? Let us go back and look at the diagram of the cell surface membrane still. The diagram of the cell surface membrane, yeah, it is right here. When you look at cholesterol, cholesterol is embedded within the phospholipids or in between the phospholipids. So cholesterol is able to maintain the mechanical stability of the cell surface membrane. How? Sometimes the membrane could be much more fluid. So if the membrane is much more fluid, usually cholesterol molecules are added. Why are they added? So that they reduce the gaps between the phospholipids. Eventually, the membrane fluidity is going to be controlled. If the membrane is much more fluid, just add more cholesterol molecules to that membrane, and eventually it's going to become less fluid. And still, if the membrane is less fluid, what you do, still reduce on the number of cholesterol molecules so that the membrane or the phospholipid get more space such that they can be much more fluid or much free to move about. So that is how cholesterol is used to maintain the mechanical stability of the cell surface membrane. So it can get in between those phospholipid molecules and reduce fluidity Or if the fluidity is too much, the cholesterol molecules can, if the fluidity is too much, the cholesterol molecules can be added to the phospholipids so that we have less movement of the phospholipids. Now, those are the major uses of cholesterol. Number one, we have mentioned cholesterol is going to be able to act as a barrier against movement of ions or polar molecules in or out of the cell through the cell surface membrane. So they will not pass through the membrane. At the same time, cholesterol maintains the mechanical stability of that cell surface membrane. It is responsible for maintaining the correct fluidity. Like an example, if the temperatures are low and the membrane is becoming so much, the phospholipids are becoming so much close together because they're not moving about so much because of a reduction in temperature. Cholesterol will prevent those phospholipids from becoming very close together because it's within them. So it will prevent them from becoming so much close together. So that is how it's going to be able to maintain the mechanical stability of the cell surface membrane. Now let's look at the glycoproteins, glycolipids, and the proteins themselves. We already know what glycoproteins are and what glycolipids are. So one of the uses of the glycolipids and glycoproteins is that they act as receptor molecules. Receptor comes from the word receive. An example, when you go to a new place usually, and maybe if you go to a new hotel, you might want to know what room you're going to sleep into, or you might want to be guided on a few things in that hotel, and the best place to go to is the reception. So even sometimes we can have molecules carrying messages, which messages are supposed to be sent into the cell. But those messages, or those molecules, most of the time attached to these glycoproteins or the glycolipids. So the glycoproteins and the glycolipids are going to be acting as receptors. They're going to be receiving those messages. An example, when hormones come in, for the cell that they really affect, those hormones most of the time will attach to those receptors. So one of the uses of the glycolipids and the glycoproteins is to act as receptors, ready to receive messages that are in form of maybe hormones or neurotransmitters. One of the other uses of the glycolipids and the glycoproteins is that they act as cell to cell, they are used in cell to cell recognition. They act as antigens, they act as cell markers. What does that mean? Still, I'll refer you back to this diagram. Now, when you look at these glycolipid chains or the glycoprotein chains, the carbohydrate chains, those chains are pointing outwards. So these chains are responsible for marking cells. So they act as antigens. I can give you an example of a student who is in school. Students usually put on uniform. So whenever someone sees you in a uniform, they will know, oh, that is a student. Now, even our cells, the different cells that are within your body, they have characteristic glycolipids and glycoproteins on their cell surface membranes. So those glycolipids and glycoproteins are used in recognition of those cells. An example that you'll learn about later in immunity, you will get to know that your immune cells, your cells of the immune system, that is the lymphocytes and the phagocytes, usually they cannot attack your own cells. How? They can recognize them by the glycolipids and the glycoproteins on their cell surface membranes. But they can be able to identify bacteria. How they identify bacteria? The bacteria will also still be identified using the glycolipids and the glycoproteins on its cell surface membrane. So that is what we mean by cell markers or cell-to-cell recognition. So the glycolipids are used in cell-to-cell recognition, or you can mention that the glycolipids and the glycoproteins act as antigens. They act as antigens. Okay, how about proteins? Now, some of the proteins are able to act as transport proteins. When you look at the proteins, they play different roles. Some of the proteins, still back to our diagram, act as transport proteins. What do they mean by transport proteins? They are going to be channels or pathways where the molecules we saw that cannot cross the cell surface membrane can pass. Like the polar molecules, they can be able to pass through those proteins either into the cell or out of the cell. So such proteins are going to be called transport proteins. We shall look at their details later. Now, some of the proteins can act as enzymes. So you find that a protein is within the cell surface membrane, but is an enzyme that catalyzes a certain reaction. So some of them are going to be enzyme in nature. Then some of the proteins in the cell surface membrane are able to be part of the cytoskeleton. Remember, we saw that within the cytoplasm, we can have there what we call microtubules. Microtubules or filaments, microtubules. Now, the microtubules form part of the cell cytoskeleton. So some of these proteins can be attached to those structures inside the cell to the microtubules. And eventually, they are going to also be contributing to the cytoskeleton and eventually maintaining the shape of the cell. Some of the proteins could still be attached to structures outside the cell surface membrane. So proteins are very important in being part of the cytoskeleton. So as a result, they maintain and determine the shape of the cell. And here they're also mentioning that some of them are going to be involved in changing of the shape of the cell as the cell moves, because some cell can move from one place to another, like the phagocytes. So some other proteins will be performing different roles in the membranes of organelles. Like we saw, the mitochondrial membrane contains proteins. So those proteins will be involved in respiration. The chloroplast membrane contains proteins, and for them, they will be involved in photosynthesis. So those are the major roles of the different parts that form up the cell surface membrane, or that make up the cell surface membrane. You have an assignment here. I want you to sit down, summarize the major functions of number 1, phospholipids, cholesterol, glycolipids, glycoproteins, and proteins in the cell surface membrane. If this video has been helpful to you, kindly give it a thumbs up, give it a like. I want that like. Kindly subscribe to my channel, share with your friends. See you again in the next one. Thank you for your time.